So firstly, welcome everyone to Pawsey Friday. This is a HPC COVID-19 Australian response showcase. We have 5 presenters who have been working on the COVID-19 response and in what I think will be a very exciting and relevant event for the times. So my name is Gadi. I'm a part of the marketing team, and I'm just going to run through a little bit of housekeeping before we get started. So first and foremost, this event is being recorded and it's going to be uploaded onto our Pawsey YouTube channel as well. So just so you know that this is all being recorded from now on. Just a little bit of Zoom etiquette as well. Please stay on mute if you can. We really encourage you to use the chat function that Zoom has. So please ask our presenters questions, ask our staff questions, and we will respond to them when we can in the Q&A section at the end of this event as well. After this event, we also have a survey. I'll paste a link to that in the chat, and I please encourage you to complete the survey as well. And should anything happen, if you drop out of the Zoom event or it goes down for some reason, there's a Microsoft Teams backup meeting that I have organized. So I'll paste the link to that if you didn't get it in your event confirmation. So if you drop out here, please join us on Microsoft Teams, but hopefully Zoom should be fine. I'll also paste some instructions on how to use Microsoft Teams if you don't actually have a Microsoft account. But other than that, yeah, I hope you all really enjoy the event. I hope it's informative for you. And I'd like to pass it on to Mark Stickles, Pawsey's Executive Director.
Thanks, Aditi, and welcome everyone to this special Pawsey Friday that we're proud to host in partnership with NCI, and you'll hear from Sean just after me. But I'd like to begin this event by acknowledging the country that we're currently meeting on. While we're meeting virtually, for all of the participants in Australia, it is our normal tradition to pay our respects to the traditional Indigenous owners of the land upon which we're meeting. So for those that are internationally sort of zooming into this, it's an Australian custom to do this. So in Perth, we're on Whadjuk country of the Noongar people in Western Australia, and so I pay our respects. I know our colleagues at NCI are on Ngunnawal country and proudly wearing, as Sean is, some of the artwork of a Ngunnawal artist as well. So we're also connected to country in the Midwest as part of the Square Kilometre Array project that we support at Pawsey. So the Wurundjeri people have a connection to what we do here at Pawsey. I also would like just to send out a note of support to all colleagues and community members that are in some places doing it tougher with respect to COVID-19 than in other parts of the country. And here in WA, we are currently managing well through what are difficult times, but our thoughts go out to those around the country who are doing it tougher and around the world. The opportunity of this showcase to get some insight into some of the frontline research work that's trying to accelerate the process of vaccine development and understand and track the development of this pandemic is something that is incredibly timely, and we're proud to support here at Pawsey. Just by way of a short update, at these regular events, we also give an update into some of the major activities at Pawsey, and we're going through a significant capital program, so an upgrade of our infrastructure at Pawsey. It's part of an Australian government commitment to both of the national centres to upgrade their infrastructure, and we're about 12 months behind the progress that NCI have made and the launch of their Gadi system. So we're on a similar trajectory to upgrade our infrastructure over the next several months. By way of a short update, we've completed the upgrade of our cloud compute infrastructure, and some of the early adopters to that were part of the process we stood up for supporting COVID-related research in partnership with NCI. So that cloud infrastructure upgrade, Nimbus, is complete. We've also installed a new cluster for our MWA, the Murchison Wide Field Array project, and that cluster is called Garawala, which is a Wurundjeri word meaning spider. So if you know the radio astronomers and their telescopes and how there are thousands of those in the desert, to the Wadri people, they look like spiders. So we've given the cluster that supports that research project the Wadri name of Garawala. We've also progressed upgrades to the ingest nodes that support ASCAP on one of the other precursor project support for the square kilometre array, and plan to install that upgrade sometime in September. And we're currently out to market for an upgrade for a long-term storage system. So it's a busy time at Pawsey with respect to the capital program and the various upgrades and activities. And importantly, we're seeking to engage in an early way with the research community to prepare them for the scaling up of our infrastructure and the advanced services that it'll offer. So last Friday, we announced a program called PACER, and information about this program is available on our website, and it invites researchers to partner with Pawsey over the next 12 months to 2 years and develop programs tackling grand challenges that are suitable for delivery at scale on extreme scale high performance computing. So with that, Aditi, I think I'll be handing over to my resplendent fellow director in Professor Sean Smith, and offering an update from NCI. But thank you all for joining us for this afternoon. Unfortunately, I can't stay for the entire event. I have a conflicting commitment, but I wish everyone well, and I look forward to watching the full event on the recording afterwards. But thanks everyone, and over to Sean.
All right, thanks, Mark. So it's how exciting to see everybody here. It's tremendous that we've got this first opportunity to get eyes on some of the early stage outcomes of the COVID-19 response research that's being undertaken at both facilities, NCI and Pawsey. I should thank federal government and NCRIS for the funding, which has allowed us to undertake our progressive upgrades. Gadi, as you will know, has been now operational in its full form since just past the new year, and is flying along really nicely. And the fact that we had that upgrade gave us, a significant amount of additional capacity. And I also then wanted to thank Mark and our friends at Pawsey for their collaborative, generous collaboration in joining to offer a joint response from Australia and HPC for research that, we hope will make a significant contribution into the broader picture of efforts to try and understand COVID-19 and find ways to effectively tackle it. Indeed, it won't just be COVID-19, because the research that gets done is going to stimulate ideas that'll head off in other directions as well. So really exciting to be able to have a chance today to see some of these early results. On the NCI and Gadi side, we've got Megan O'Mara from ANU presenting some results of work that she's been doing on Gadi. And I thought that Megan, I think, is also resplendent in some Gadi merch. We've got Shelley Barfoot from Alan Mark's group up at UQ. So looking forward to it. I won't take up any more time. Oh, except one very important update. NCI is going to have more Gadi merch. So watch this space. You'll be able to get some socks soon. How about that?
Thanks, Sean. Okay, I'd like to introduce Mark Gray to everyone. He is going to be helping me host today's event. Mark is Pawsey's Head of Scientific Platforms, and he's just going to begin with a bit of an overview on the COVID response and then introduce our speakers. Over to you, Mark.
Thanks, Didi, and thanks to Sean and Mark for introducing the centre. Sean, I'm really looking forward to some socks from Gadi. That'd be great. So welcome everybody to the HPC COVID-19 Australian Response Showcase. We're going to hear from several researchers that are using Australia's peak facilities. In early April, Australia's Tier 1 supercomputing facilities, Pawsey Supercomputing Centre and the National Computational Infrastructure joined forces to offer additional computation and data resources to support national and international research community efforts to acquire, process, analyse, store and share data supporting COVID-19 research. Both facilities contributed extensive resources to assist researchers in the fight to overcome COVID-19, And through this initiative, researchers throughout Australia are now working with the NCI, which is currently supporting 3 targeted projects with more than 40 million units of compute time on the Gadi supercomputer, and Pawsey Supercomputing Centre, which provides access across 5 projects to over 1,100 cores on the newly deployed Nimbus cloud. During this event, you will hear updates from hands-on researchers working on understanding the lineage of the virus, making statistical inference about potential treatment outcomes, COVID-19 targets, molecular modelling, and more. First, I'd like to introduce Dr. Gareth Price, who's the head of computational biology at the QCIF facility Advanced Bioinformatics. Gareth manages the diverse spectrum of researcher-led questions involving genomic data, provides training in genomic data analysis, as well as leading the Galaxy Australia as the platform manager. Gareth has 20 years experience as a bioinformatician and genomic scientist, and he's going to talk to us today about the Galaxy Australia COVID-19 decorated pulsar node. Over to you, Gareth.
Thank you, Mark, and thank you, everyone. I will do my best to keep to time. Please keep me to time. Right, so Galaxy Australia is going to be a slight anomaly, I think, in today's response. We've looked at enabling the global response to COVID-19 research and insight through enabling an open source platform we have, which is Galaxy Australia. Very briefly, we want to thank QCIF and Melbourne Bioinformatics and UQRCC for maintaining the national platform, and BPA, ARDC, the Biocommons, and NCRIS for their ongoing support and commitment. For those of you that don't know, Galaxy Australia is a hosted web accessible platform that lets you conduct accessible, reproducible, and transparent computational biology research. What that all translates to is it's a website. And its website is usegalaxy.org.au. And of course, we have a Twitter handle as well for those people that want to follow us there. The website has this style of format. Tools down the left-hand side, we have over 950 tools now for all manner of life science analysis. Used to be a core basis of genomics analysis, but it is now increasingly proteomics, metagenomics, and metabolomics, and it will continue to grow. The right-hand side of the screen is your history in the Galaxy term, which is effectively an electronic lab book for each of your data sets. The real power of Galaxy, and what we want to highlight today, is that you can set up a workflow. You can chain tools together that are executed once to do a complex series of analyses. In this case, taking raw sequencing reads, mapping against a database, creating a reference, converting those into a particular format for visualization and plotting. And all that can be managed in version controls so that it really emphasizes that reproducible science. In terms of the platform, again, just to emphasize, it has, as I said, there are over 900 tools. Of some of the visualizations one can perform are on the right-hand side of the screen. So there's some wonderful chronoplots for bacterial diversity or sequencing depth along a genome as visualized on the bottom right. Whilst we are very proud to host over 900 tools, we actually have access to over 7,000 tools on a global repository, and I'm going to continue to emphasize the role that Galaxy Australia plays globally in e-research and life science, but also the role that the globals play in helping us perform that research here in Australia. And all aspects of the platform are completely peer reviewable and transparent. Nothing's hidden because we want people to publish on our service, and we're thrilled that they do. In terms of what we actually did, so that's the brief, is encompassed in this preprint, which is now in actually draft print on PLOS pathogens. Under the title, No More Business as Usual, Agile and Effective Responses to Emerging Pathogen Threats Require Open Data and Open Analytics. A joint paper from Galaxy Europe, Galaxy Australia, Galaxy Belgium, Galaxy Maine, of which Australia took the handle in running the genomics. So the assembly, the timing events for mutation, the variant analysis, and selection and recombination identification was handled on the Pawsey machine. This formed only part of a greater suite of tools that were rapidly generated and deployed globally, evolution-based tools, chemoinformatics tools, proteomics, Amplicon-based data analysis, which was quite fascinating and is also now available on Galaxy Australia since the initial release of these 6 workflows, and direct RNA-seq analysis. If you would like to know more, all this information is housed at covid19galaxyproject.org. What it actually looks like mechanistically, and I realize for many people, unless you've got about a 70-inch screen, these workflows are going to be unreadable. What this is really to emphasize here is that workflow nature of the platform. So whilst it's a graphical user interface, you can interact on your computer, on your phone, on your tablet, on your TV, through a web browser. Really, it's a workflow management system, and the workflows we have highlighted here are the pre-processing assembly, the MRCA timing events, the variation analysis, the sequence analysis, and the evolutionary analysis are all chained workflow events to assemble raw sequencing data into some understanding of the evolution and variation you're experiencing in your SARS-CoV-2 cohorts. So, on the basis we don't all have massive screens, I have just zoomed in on one of those. It's the variant analysis workflow, just to show you the sort of complex nature of parsing of data we can do in Galaxy, both as individual files or collections, from the left-hand side of our source material through to our collapsed collection on the right-hand side. The way Galaxy works, everything that started with a little green star is an effective output for the user to interpret. Everything in black is actually produced, just pushed into the background to summarize and make your reviewing of your data a little easier. Now, Pawsey and Galaxy. The real beauty of Galaxy as it's designed is it's a single web interface that needs to be physically hosted somewhere. It is hosted at QCIF, enabling on the Chris Cloud technology. But the head node can schedule jobs to any remote pulsar, and we're very proud to be running 2 pulsars in Melbourne at Melbourne University through Melbourne Bioinformatics, and with the call for the COVID release, we were rapidly able to deploy a pulsar onto Pawsey. And the real beauty of it is that through that head node configuration, we can send jobs that we want to each pulsar location. In this case, we schedule any job teed to a COVID-19 workflow, which I'll show shortly, to the Pawsey Nimbus cloud, meaning that your COVID jobs are not getting clogged up with all the other legitimate researcher jobs on the Melbourne 2 Pawseys or on the head node. They get dedicated access time on Nimbus and are quickly returned as results to the researcher.
5 minutes to go.
Thank you. As I just wanted to emphasize again, but mentioned earlier, Galaxy Australia does this in conjunction with Galaxy Europe and Galaxy Main in America. And by does this, I don't mean we are sharing user data, but we're sharing the workflows and the tools and the reference files and the configuration styles that support this rapid analysis on each of those services. So to actually get to what we did, for those people that wanted to know the technical details, what we actually deployed was a pulsar system. A pulsar system in this case contains a head node, and in the operational capacity here at Pawsey, we also deployed 5 dedicated worker nodes, all of them as an 8 CPU, 32 RAM flavor. So how we did that, very broadly, we applied for the merit allocation. So again, thank you to Pawsey and NCI for enabling that. We set up the virtual machines, which was 6 virtual machines, the 1+5 is described there. What we're very proud of and what allowed us to do this extremely rapidly is that the configuration for each of those head node and worker machines has been put into Ansible. A simple matter of running those Ansible scripts to deploy a completely functional Galaxy system, Galaxy Pulsar system in this case, onto those VMs. We linked that in with our CERN virtual machine and file management system. It's a tier 1 we have in Australia, which maps in all the appropriate reference files. And the last thing we do, which is, it sounds like internal housekeeping, but I've already mentioned it, it's very important. We then go through and configure Galaxy Australia, which has a tool destination matrix to reflect the new destination, in this case Pawsey, and configure it so that all the Galaxy COVID-19 workflows were directed to Pawsey. So in terms of rapid, we were quite proud of that. That was about 2 business days to do all that work, spread over a little bit of time. Well, no, actually, literally 2 business days to get that up and running, to have all our workflows available for people to run. What we do have on our landing page is a clear direction for users of the platform, and currently we have just shy of 9,000 registered users on Galaxy Australia. References to the learning portal at Galaxy Project. We have shared workflows, and I'll show those shortly. If you want searches for COVID, we have shared reference data. And by shared, we mean this is publicly available, easily ingestible into your instance of Galaxy, and reinterpretable or to be plugged into your research. At this global website, training.galaxyproject.org, there are also 3 tutorials with a hands-on set of slides, publicly available data, workflows, and a link to which global resources these can be run on. And there are 3 current workflows we have to enable COVID-19 or SARS-CoV-2, more accurately, analysis of the genome and variants, and some of the cheminformatics as well. What we have on Galaxy Australia is this series of workflows. It's a minor detail, but if you look at the right-hand side, you can see we had a bulk upload of these in February to capture the genomics aspects of analysing SARS-CoV-2 genomes. So again, the 6 steps that I showed on the slides earlier. And then more recently, in the last month or so, we have added all the amplicon-based SARS-CoV-2 analyses workflows.
10 minutes.
Great, last slide. So again, thank you to Pawsey and NCI. What we can say for all that work is we know we have enabled the analysis of hundreds of SARS-CoV-2 genomes, amounting to over 9,000 Galaxy tool executions on Nimbus since February. And we're very proud of enabling that support, that research around Australia. Thank you very much.
Thanks very much, Gareth, for all that. That was very informative. We're going to actually, at the end of all the presentations, have a panel discussion around this. So please save your questions for the end. There'll be opportunity to share those and get answers from the panel members, which are all the speakers that are talking today. I'd now like to switch to hearing from Associate Professor Megan O'Mara from the Australian National University. Megan studied medical science and worked in pathology labs before returning to university to study physics. Her research uses computer simulations to understand how proteins in the cell membrane act as molecular machines to transmit information across the membrane, and how we can design more effective pharmaceutical drugs to treat conditions such as chronic pain, chemotherapy resistance, antibiotic resistance, and more recently, COVID-19. So over to you, Professor O'Mara.
Thanks a lot for that, Gareth. Sorry, Mark and Gareth. I would like to start by saying, in order to have a drug or a vaccine that effectively targets a problem, we first need to know whereabouts in the body that problem is, and then we need to know the machinery that goes into how everything works so we can target that effectively to stop it from happening. So with that, I'd like to firstly acknowledge my group, in particular, Dr. Katie Wilson, who you can see here, Dr. Stephen Fairweather, who is down the bottom here, and thanks to the wonders of Zoom, a lot of this work was done over Zoom, and also NCI and Pawsey for their support. I've got a couple of experimental collaborators in here, Associate Professor Malcolm McLeod, who is a medicinal chemist, and Professor Stefan Bra from both from ANU, and Stefan is a biochemist who's interested in transport proteins. So with that, I'll start by sharing with you this image I got from a website, and everyone knows what this is. It is coronavirus. It's an artist's impression of coronavirus, And generally we see these big spikes and we go, oh, this is pretty scary. What we're interested in my group is how coronavirus actually binds to the human receptor. And we'll focus in on where the spike binds to the receptor of the human cell and what happens. So if we zoom into this, we can see in more detail another artist's impression of this spike protein of the virus binding to a human receptor that's embedded in a cell membrane. So once the spike protein binds to the receptor, all sorts of conformational changes and things happen in there. And what we see, I've gone backwards instead of forward, sorry, is that the virus can then fuse with the cell membrane and empty its genetic cargo into the cell to hijack the cell and produce more copies of the virus. So what we want to know is actually how the virus interacts with its receptor. So the protein structure of the receptor has been solved. And what we know is it's one molecule of this protein here called ACE2, and I've got a cartoon image of it, and it's in complex with another protein called BOAT1. And the spike protein from the virus binds to ACE2, and then all sorts of things happen, and this allows the virus to enter into the cell. You have to have binding of the viral spike protein to ACE2 in order to initiate this. So that's great, but that's not the whole story for this receptor. Actually, when we look at the structure of the receptor, we have 2 copies of BOAT and 2 copies of ACE, and they can bind to 2 copies of the coronavirus spike protein. So what we were really interested in is what happens to BOAT and what's going on there, because a lot of people are looking at ACE. So BOAT is a transport protein in the cell membrane, and what it does is it transports a cargo. In this case, the cargo is amino acids into the cell. So the amino acid or cargo comes along, it binds into a particular cargo bay in BOAT. There is a lid on the cargo bay. It's in an open state here while things go into bind, and then it closes. And so then we have a closed state for BOAT. So this affects the overall receptor, and what we want to know is how does this all work? How does the BOAT1 ACE2 complex bind the viral spike? We also want to know, does the BOAT1 ACE2 receptor complex behave differently when BOAT is open or closed? And can we use an alternative cargo for BOAT, like a drug, to prevent the virus from infecting the cell? So what we do is we use a technique called molecular dynamics simulations. These basically use Newton's equations of motion, the same ones you learnt in high school physics, to track the motion of the protein, all the water molecules, the cell membrane, and all parts of the system over time. In our system, there's about 800,000 atoms that we're tracing, and so we really need the supercomputer in order to do this. So
5 minutes.
Okay, I'll hurry up. So what we did is we did simulations in the open and closed state, with and without cargo, and with a drug instead of the cargo. What I'd like to show you now is a movie of our simulations, starting from the closed conformation of BOAT and also one in the open conformation of BOAT. What you can see as the simulations play through is that the complex with the viral spike, ACE2 and BOAT, move in very different ways in the closed conformation and the open conformation. In the open conformation, you can see the motion of ACE2 and the viral spike protein is much greater and in the opposite direction to the closed conformation. So one thing we can do with this is we can actually produce a kind of representation of that motion as this thing called an RMSD. You don't have to worry about it too much, it's just a representation of the motion. And we can see that BOAT moves the least. And this is the open conformation, and these ones represent the closed conformation. of these, ACE2 moves an intermediate amount and the viral spike protein moves the most. So we can see that when boat is open, the cargo bay is open, then we have a lot of motion. When the cargo bay is closed, we have a small amount of motion. And we can actually drill down into the details of this. If we look at the surface of the proteins, we're just drilling in on ACE2 and the viral spike binding area. And we can see that the surface of those proteins look different in the open and closed conformation. And in the open conformation, you can actually see small spaces between the viral surface and the surface of ACE. If we then go into the chemical details of those, then we can pull out the details of the interactions. And we find that in the closed conformation, we actually have 7 hydrogen bonds formed at this interface between the viral spike protein and ACE. And these hydrogen bonds persist for about half the simulation. In the open conformation, we have much less chemical interactions. We only have 2 hydrogen bonds, and they only stay for about 39% of the simulation. So the other thing we can do is see what happens when we put some cargo into boat. So we can either put the amino acid that's meant to be in there, the natural cargo, or we can put in a drug instead. So when we do this, we put this into the drug binding site in boat, which is in the middle of the protein here. You can't actually see whether the cargo bay is open or closed in these simulations because this is a chemical detail right in the center of the protein. It's not visible from the surface. So when we do these and we run these simulations, we find that we've got the open conformation here as a comparison and the closed state with the cargo in there, this is the amino acid cargo, has a much smaller motion overall. With our drug compound here, it also binds at the same place that the native cargo, the amino acid, binds. And we can see that this also decreases the motion of the whole complex here. So we find that when a drug is bound, then we have much less motion in the complex. And what we're hoping is that we can get this drug to block the motion, block transport through boat so that it never opens. And this will stop the motion needed for the virus to fuse with the cell. So in conclusion, we find that overall the motion of the boat 1 ACE and the viral spike protein is inhibited by having a closed state of this receptor, so that cargo door closed. If we add the cargo, the amino acid, to boat, then it further inhibits the motion. And if we use this information to design drugs, then we may be able to produce a drug that targets boat to prevent the entry of SARS-CoV-2 into the cell. Thank you for your time.
I'd like to now introduce Tom Karagiannis from Monash University. Tom's PhD studies were aimed at developing DNA-targeted cancer therapies, and these were continued during postdoctoral studies in the Molecular Radiation Biology Laboratory. Tom headed the Epigenomic Medical Laboratory at the Alfred Medical Research and Education Precinct, the Baker Heart and Diabetes Institute, and is currently at Monash University. In addition, he's been involved in teaching at the Department of Pathology at the University of Melbourne. Over to you, Tom.
Thank you, Mark. And thank you for the opportunity. I'll just share my screen. Here we go. Okay. Yep. Everyone can see my slides? Good. Good. Okay. Thanks very much, Mark, and thank you for the opportunity. I'm going to talk about the main protease today. So it follows on nicely from Megan's talk. Megan talked about the initial infection with the ACE, binding to the ACE receptors. But I'm going to talk about the main protease and how we can potentially inhibit the main protease. So my lab is based in Melbourne at the Alfred Centre, and I've lost control of my slides. I got it. Good. Yeah. Excellent. So this is a picture of my lab here. So the key people is Catherine. Catherine does the wet lab, so she validates our findings. And Julia, you'll see more of Julia in a minute as well. So Julia's an all-rounder. She's part of the lab and part of the simulation team as well. So usually we perform our experiments in this building here in the city campus at RMIT. And we do that in collaboration with Dr. Andrew Hunt. at the RMIT University. As you can see, there's no one on campus these days. And also we go to Starbucks and that's closed as well. Anyways, so the team, as I said, Dr. Andrew Hung, a good collaborator, we've been hacking around for about 10 years now. Eleni Pitsiou is a very competent master's student. And Julia Lang again, the girl that I showed earlier. And this is the protease, the main protease that I will be focusing my talk on. So you'll see more of this. This is the actual protease that we try to inhibit. There's also Kevin, part of the team, and Chris. So if you can notice the surname here, if you're astute, it's the same surname as mine. He's my son. He's my son. He actually he's fresh. He's fresh. He majored in virology, but so he thinks he's a resident expert. I won't bag him anymore because then he might be listening and then we'll have problems. So the way we went about getting into this is we've got our own chemical library, and as I said, we've been hacking away with molecular modeling with Andrew for about 10 years now, and we have our own chemical library that we can investigate. And again, this shows the different compounds that we have. They're based on the olive-phenolic compounds. Okay, so I'll get into what we do. And again, I'd like to thank Topaz because we wouldn't have had time to do this, Pawsey and Topaz actually, for giving us 200,000 service units of advanced GPU clusters. And I'd like to thank Audrey because she coordinated all that for us. And then Karina and Aditi, I like getting their emails. I enjoy opening their emails more than I do finance from our department. So they're always happy emails. And of course, our computer specialist is Marco Delapierre, and he helps the girls all the time. He helps Julia and Eleni with their modeling requirements. As I said, in our first quarter, so just to get a feel for the allocation that we have, in our first quarter, we had enough simulations for 3 publications, for our first 3 publications. So if you think about our system, this is the entire system here. This is our protease that we model, and it's about 133,000 atoms. So the time that we've been allocated, in the first quarter, we'd be able to run about 4 to 5 microseconds of a simulation, which is not bad. Okay, so as I said, Megan talked about the initial fusion of the spike protein to the ACE2 receptor here. And what happens is that the virus gets internalized, it fuses with the cell, gets internalized. And we're interested here because what the virus needs to do is it needs to replicate. And in order to replicate, it makes copies of its own genome, of course, and also it makes a lot of proteins. And these proteins are long polypeptides, and they need to be cleaved somehow. And the enzymes that we're interested in, so the proteases that we're interested in are responsible for cleaving the polypeptides into the right size. So then the virus can repackage itself. It hijacks our Golgi apparatus. It forms another copy of itself, and then it gets released from the cells. Once it gets released, it can then infect other cells in our body, or we cough it out, or sneeze, and then we have to socially distance and wear masks and buy lots of toilet paper. So the main protease is this one here, as we said, and a lot of people, what they're trying to do these days, as everyone is aware, is to repurpose drugs that are already available, because that's the easiest path to the clinic. Okay, is repurposing drugs, and one example is lopinavir over here. So lopinavir and ritonavir. These are anti-HIV antivirals, and they've also been shown to bind in the binding pocket of our protease here. Okay, so that's one of the angles that people are trying to do. So I'll show you guys what we're trying to do. So in our first paper, just to start off with, we modeled an alpha ketoamide 13B. And these kind of ketoamides, they're the prototypical inhibitors, right? So this is the inhibitor here sitting in our protease. And the reason we started with that one there is that there's a group out in Germany, so headed by this guy here, and they're trying to make structural analogs of this compound in order to make it pharmacokinetically, better pharmacokinetically, so it can be used in humans. So our very first paper we published is this paper here, where we looked at the active site of the main protease. Okay, so nice and simple. And what we found is we're one of the first to confirm that yes, this ligand does bind very tightly in the active site of the main protease. However, as with all science, it gets a little bit more complicated, right? So one of the big problems is that this protease comes with two monomers, right? So this is a monomer. This is one part of this here. Okay, one part. And
5 minutes.
5 minutes? Good. So this is one monomer, right? And what happens is, until March the 30th of this year, only the monomer was available on the PDB for modeling, okay? So this is a perfect example of some science, right? Where the science is, I'm not gonna say it's wrong, but it's been perpetuated and there's a little bit of inaccuracy, and I'll show you why. What happens is that when we dock ligands to this monomer, as you can see here, these are the ligands in yellow, they always go to the active site, right? So the active sites, a lot of the compounds go to the active site. But what we find is that, and even when we do our modeling, what we find is that approximately 50% of the ligands, right, are binding in this active site. Now this can't be right, especially when we look at massive modeling efforts. So as I'll show you later, some people have done like 1.3 billion compounds, right? And 50% of those are binding in the active site. That's kind of ridiculous, right? Because then you're getting like a large, like 800 million actually going to potentially active. So what we did is we did some site mapping of the dimer. So here, what we've done is we've taken the monomer. This was what was usually available. A lot of the compounds are going to the active site. Okay, that's good. When we look at the dimer, so if you look at the dimer here, what we found immediately was that this is really highly reactive binding pocket at the apex. Okay, and when we do blind docking to this dimer now, what we find in this case, these are red now, these are the ligands in red. Most of our ligands are going to this apex, okay? And not many are binding in the active site. And this is the basis of our second paper that we're publishing now. So our second paper is looking at the site mapping, and we've used a couple of different programs in order to verify that this is true. And we always come up with this active site here, okay? An active site at the apex of the dimer. And what we find is when we model our compounds, a lot of them are binding. So it's like a sink. It's pulling all the ligand towards that area and away from the active site. So the active site is shown here in brown, and the ligands here in pink are up the top here, right? So what we've got is we've got a highly reactive active site, and it's taking away a lot of ligand. So that's the basis of our second paper. Having characterized the active site now and potential allosteric sites, like this site here, our third paper is where we're actually looking for hits, for compounds that we can actually say that are going to the active site and actually can be useful, okay? And as I said, we've got a lot of competition, and in this case here, as I said earlier, one particular group, they looked at 1.3 billion compounds. So there was no way we can compete with 1.3 billion compounds, so I had to look back in history. So I looked back in history, and I am Spartan, so I thought we have to do 300. So that's why there's 300 up here, right? So we're looking at 300 compounds. And what we've done is we've modeled those 300 compounds, and we modeled them using two different programs. One of them is commercial, one's a free program, and we get quite good specificity. So that's where we're at now. We've sort of published 2 papers already. We're on our 3rd paper. And of course, again, I'd like to thank Topaz for giving us the time, otherwise we wouldn't have had any time to do any of this work. Thank you to Aditi, Karina, of course, and Marco, and of course to Andrew Hung and my students here, Lenny and Julia. So thanks very much, everyone. That's all.
Tom, thanks very much for that. Again, I guess a reminder of the way the fundamental research in chemistry and physics is tying into getting at the answer to what's actually happening at the molecular level as this disease is trying to infect our cells. I'd like to now introduce Associate Professor Michael Wise from the University of Western Australia. Michael is a computer scientist working in computational biology since the early 1990s. His early contributions related to in silico investigations ranging from the plant desiccation tolerance protein LEA to low complexity and natively unfolded proteins. For the last several years, he is mostly involved with microbial informatics and is particularly interested in the evolutionary biology of microbial species and viruses. So over to you, Professor Wise.
Okay, so this is now zooming out from those really two nice mechanism-related talks, and which will actually, as you'll see later, set me up rather nicely. Because I want to look at the evolutionary biology of the new variant SARS. As it happened, this whole happened whilst I was supposed to be on long service leave, but I couldn't go anywhere suddenly. So I sat down and said, well, what can I do? Oh yeah, right, let's do some evolutionary biology of SARS. So here is the coronavirus. It's basically a beta coronavirus. It's a positive sense, single-stranded RNA virus. It's quite long for these sorts of viral genomes. It has 28 genes. And as Tom said, the first bunch of them end up being translated into a single polyprotein, which is then translationally cleaved. It turns out that across these beta coronaviruses, the gene order is very highly conserved. And the word conservation is now going to turn up actually a lot. So what we know is that the expert committee found that the new SARS and the old SARS are essentially sister clades. In other words, they're very, very closely related. But what they did was they used only 3 genes to do that. And I wanted to use a bit more data than that. They used the genomic data, and I wanted to use protein, because although change very obviously happens at the DNA level, evolution works at the protein level. In other words, it's the interaction between the proteins of the virus, ultimately, and the proteins of your body. That's what really matters. So the first thing I want to ask myself is, well, how well conserved is this particular virus? Because I've previously done some work on HIV and influenza, and as we all know, they change a lot. So I wrote a program in 2013 which I called Mean Protein Evolutionary Distance, or MEPED. And what it's trying to measure is what's the evolutionary pressure on a virus, particularly on the viral proteins, because the host immune system is forcing the virus to change. But the proteins don't want to change because they suddenly stop working. It's a bit like spinning a rope. There's the 2 different sorts of forces acting, and this was kind of what's happening here. So I started with the 26 proteins from 1400 SARS covariant to new SARS genomes. I extracted the translations, the protein translations from the genomes, and then I built a tree per protein. So that's therefore 26 trees. I used a fairly ancient tree builder called PhiML. I mean, I would use something now, but I used that so I could look at the values and keep consistent data with what I'd done earlier. And so the way I did it was basically blacksmith parallelism, really. I just put an instance on each CPU on Nimbus. So I just did a make. If I were to do it again, I would use FD, which I've just discovered recently. It looks rather cool, where you can spawn processes across a bunch of resources. So that's maybe worth another look. The long and the short of this story is that the proteins are ridiculously conserved. Very highly conserved at the protein level. So although there's a lot of discussion in the literature and on news about how it's changing, there is a lot of change because it's an RNA genome, at the protein level, it really stays. It's so conserved that it looks like dengue, and it's actually a similar sort of virus to dengue. And it's even as conserved, and what's even more conserved than dengue, it's almost as conservative as measles, which is a double-stranded DNA virus, which are traditionally very, very conserved. So I then wanted to create a phylogenetic tree. I used a tree builder called Beast, which is a Markov chain Monte Carlo. Markov chain Monte Carlo is inherently ridiculously parallel. And so the other one was working on the CPU instance of Nimbus. I was using the GPU, and it turned out to be about 10 times faster, which it was love at first sight. This was just stunning. The thing just ran. And so I was working with 58 SARS to, 58 new SARS to all old SARS, 12 bat coronaviruses, and 2 from civets, because you recall that with SARS, whilst the bats were distal to the pre-evolutionary process, the civets were actually proximal. It was
5 minutes.
It was 25. Sorry.
5 minutes.
Right. So there was 25 proteins in each concatenation, 10,554 amino acids. First, you compute the parameters in any of these simulations, and that's a whole story, which you can see in the paper. There's a draft paper. The mutation model, the clock model, but more interestingly was the population model. In other words, will the population look more like a coalescent model, which means it's all the same species and it's just broadening out, or is it a speciation model? In other words, we're now forming different species. Long and the short of the story of that is that it's a coalescent model, same species. But we know that that's what the expert committee found. Right, but I was, that of itself was kind of interesting. But then I said, okay, let's chuck in some MERS, because things were still very, very similar. Let's push out the envelope. So I found 15 MERS, which is the Middle East respiratory syndrome, from human, 7 which were relatedly from camel, 14 from bat, and 1 from hedgehog, believe it or not. This time it was 24 proteins, and that was the length. And I used the same modeling process, get the parameters, yada, yada. But in particular, when I was modeling the population, amazingly, all of the population models ended up with exactly the same log likelihood. Once you simply just got it to the nearest integer. They were slightly different, but they all ended up at the same number, which I thought was absolutely amazing. So then I thought, well, let's throw them all together, the MERS and the SARS. So this time there was 19 proteins that they all had in common. So still really highly conserved, right? 9,800 amino acids. And blow me down, this was totally unexpected. What I got as the most likely model was once again a coalescent model. Now you would think that MERS and SARS would be different species, but from an evolutionary point of view, yes, they bind to different receptors, and so it gets me into perpetual arguments with real virologists. But from an evolutionary point of view, these are really the same species, and that to me was totally unexpected. So then I did so here I've done a dated phylogeny. So the green is the new SARS, the dark blue is the old SARS, the cyan is the civet. You can see that the most recent bat split off from the most recent split from a bat sequence was 2012. So this, and what you also notice is that the progenitor of new SARS has been running around with the progenitor of old SARS at pretty much the same time. So the next trip was to let's do a grand tree out of all of them. And this was seriously large number of computations. So here is the SARS data that you saw earlier. Here is the MERS. Now, keep an eye out for the lilac strains over here, which are the bat ones, and the brown ones over here. Bat, bat. And if you trace it forward, what you end up seeing is that it ends up, the common theme is bat in both cases. So in other words, there's a circulating bat virus, which has been pushing into humans a couple times.
1 minute.
So the last thing I did was did a population estimation, which is a skyline called Bayesian skyline. And essentially what you're looking here is, this is the population of unique individuals across quite a long space of time. And essentially it's static. In other words, this is the same set of species that have been running around, and essentially it's the same genetic material until there's a crash around 2000, it began 2009, hit a minimum in 2019, and then has spiked up. In other words, we're seeing a population bottleneck which has given rise to the new SARS. That's what we're looking at here. In other words, this is quite an old story. This is not something that invented in a lab somewhere. This has actually been happening for quite some time previous to this. So where am I going now? I want to now, having pushed the envelope quite far already, I now want to push it even further and add the beta coronaviruses, which cause the common cold, 20% of those symptoms, called HKU1 or OC43. And the other thing is the SARS is the new variant SARS is actually quite recombinant. So I want to actually deal finally with recombination, because most of the previous analyses really haven't, and look at the impact of recombination. It's actually much less than you think, but it does exist. Okay, and I'm done. Excuse the awful pun at the end.
You don't have to excuse the awful puns, Michael. They're always welcome. So thank you very much for that, and for the big picture view of what's happening with this virus and kind of where it comes from. We're now going to go to our last talk. And this talk is from Shelley Barfoot from the University of Queensland. Shelley Barfoot received her bachelor's in microbiology and computer science through the advanced study program in science with honours in computational biology from the University of Queensland. She started as a PhD student in Alan Mark's group at the School of Chemistry and Molecular Biosciences in 2018. In 2019, she was nominated for a Future Superstars Award and represented the school in the 3-minute thesis competition. Shelley is the head tutor of Theory and Practice in Science, one of the foundation mathematics and programming subjects for all first-year science students at UQ. Her research primarily focuses on the understanding how viruses interact with human cells, a mechanism that is not well understood. When not researching or teaching, she is pursuing a degree in communication, playing Magic the Gathering, or gradually dyeing her hair every colour of the rainbow. Shelley, over to you.
Thank you. So it's only through years of dedicated work that we've been able to respond effectively to this pandemic. I'm privileged today to be representing the group of Professor Alan Mark to be talking about some of the work that we've been doing in response to the pandemic. This talk will be split into 2 main sections. Firstly, about understanding viral fusion, and secondly, some work we've been doing on the automated topology builder. So to start off with, you've heard a lot about the interactions of the virus with various cell components. So I will be a bit brief here, but essentially we're looking at these spike proteins as well. So these little tripods that sit on the outside of the viral cell, and how they interact with not the receptors, but the actual membrane itself, to try and understand, well, if we can stop these proteins, we can stop the virus entering the cell and in turn causing disease. So the question here really being, how does the spike protein assist the virus in invading cells? So you can see here part of the viral life cycle for most envelope viruses. So you've got it interacting on the outside of the cell with these receptors. And then the bit that we're focusing on is within this endosome. So inside the endosome, these proteins are subject to low pH, which is the human cell trying to kill the virus. But instead of killing the virus, what this does is it causes these little tripod proteins to undergo a conformational change. And due to this structural change, there is a fusion event between the viral membrane and the host membrane, which allows this virus to enter the cell and cause this disease. You can see on the right-hand side, this is from the SARS-1 virus, a bunch of these viral cells inside the endosomes. So, as mentioned in my bio, my focus of my PhD is looking at these viral fusion proteins. So there was already a significant amount of foundation work done on the understanding of this before we even jumped into the case of SARS coronavirus 2. So here we have the case of Ebola virus, which is another class 1 viral fusion protein. You can see this is a single fusion protein in its active state. So this is when it's been subject to low pH, it's interacting on quite a planar surface to both the host membrane at the top and the viral membrane at the bottom. So it's got quite a big surface there. And then what has to happen is we know in the post-fusion form that the pink regions here line up with the red regions in what's called a 6 helical bundle. So this conformational change is what drives these two membranes to come together. But in the case of COVID-19, we really want to get an understanding of how this is happening. So we want to understand this conformational change, because you can imagine if we could trigger the conformational change before they're proximal to a membrane, this would stop the virus entering the cell. Or if we could stop the conformational change happening at all, well, the virus couldn't enter the cell at all, couldn't cause this disease. So it's a really attractive vaccine target for this reason. We're seeing more and more structural data being released every day, but we still don't have a complete resolution of what this fusion protein looks like, particularly inside the membrane, because that's a really difficult experimental problem. But the part of the fusion protein that's actually interacting with the membrane, so the end of the S2 region, we have begun simulating using the time on the NCI computers. So we know that this form of the fusion protein is going to turn into a more elongated form, like you can see on the right-hand side from a cryo-EM structure. This is still quite a short simulation. So this one is 850 nanoseconds. You can see there's a fair bit of wiggling, but we're not seeing any structural rearrangements. But if we can look at the dynamics further and try and better understand what's happening here in terms of the elongation, then we can get a good idea of where we might want to be targeting to stop the process. So I said this talk was in 2 parts. I've told you about one attractive vaccine target, but obviously there are many different possible drug targets for COVID-19, as you've been hearing about today. So there are 26 viral proteins that are needed for replication, and you can see on the right-hand side a bunch of these. If we just zoom in on a specific one for an example, we can see here the polymerase is obviously an important part of the viral life cycle. So another thing that we could potentially target with antivirals in order to stop viral replication. As you've also heard, a really attractive thing to do is to use chemicals that have already passed their safety trials, so their phase 2 trials, because these can be effectively immediately brought into market. So what we want to do is we want to test the effectiveness of these. An example here is remdesivir, which has been used previously to target polymerases in viruses. So originally conceived for hepatitis C, this virus, this drug effectively stops the polymerase. And it has been tested with a couple of others. So it was tested on the original SARS and as well as Ebola, and hasn't shown to be effective on them. But this is a really interesting drug because it was shown to be effective using computational drug design. So it does prove that that is an effective method. So in order to do computational drug design, what we really need is good parameters of our target molecule, because if we don't have good parameters, then when people are doing their docking studies and such, obviously it's not going to be a realistic representation. So this parameterization is a really important step. And given that so many groups are shifting their focus into computational drug design, having an effective resource of these parameters is super important. And that's where the automated topology builder comes in. So it provides high quality atomic interaction parameters for use in not only structural based drug designs, but other functions as well. Using quantum mechanical calculations, we can provide accurate parameters so that people can then use these later on when they are doing these structural based drug designs, so doing their docking and such.
4 minutes.
So the ATB is widely used all over the world. You can see that our main user base is actually in the United States from the last 12 months. And we have over 400,000 pre-calculated molecules, which is really great to see. But the reason that the ATB is really special for this kind of work is that most databases, so you saw before there was an example from Campbell for remdesivir, they only contain the drug in a single form, whereas most drugs actually exist in multiple forms, multiple tautomeric forms in solution. So you can see here the example of warfarin. There are many different forms of this drug. You'll notice that on the right-hand side, a lot of them look quite different to each other. So you can imagine that the way that they interact is quite different. And only one of these forms is an anticoagulant. All these other forms have different functions. So it's important not only that we have good parameters for a single structural form, but for all the forms of a given drug, which is what is possible with the ATB. So using the ATB, we can parameterize all of these phase 2 molecules. So there are about 8,000 molecules which have passed phase 2 clinical trials, and we have so far parameterized about 4,000 of them. And then from there to parameterize all the tautomeric as well as protomeric forms of these molecules so that we can get a good idea for the different forms that they're in and how these may interact differently. And then finally, the aim there is to provide a rank optimized low energy structures for use in docking. So to really assist the great portion of this work that is being done there. And the work done on the ATB could not have been possible if it weren't for the significant amount of time that's been put into it. So not only in the pandemic currently has a significant amount of hours being put into it, the NCI being used for a lot of these phase 2 clinical trials. The Pawsey systems have been really useful because we don't have a wall time limit on there. So that really allows us to parameterize some of these larger molecules. But the fact that we have been working consistently on this for the past 10 years is really what allowed us to make sure that we would have the best parameters available. It is a free to use service, so it's really creating this database for the best science to come off it. And we believe if you're going to do something, you would do it once and you would do it well. So we're making sure that everything there is available and effective for use. And on that note, I would like to thank the other people involved in this work, Alan and Martin, as well as the entirety of the MZ group, as well as obviously Pawsey and MCI for not only the opportunity to present this work, but for all the computer time they've given to us now and over the years.
Great, thank you very much, Shelley. And I guess a really elegant demonstration of the value of long-term investment in the scientific enterprise.
Cool, okay, so we might get into a panel discussion based on some questions that we have. So we've got a couple from the audience already, which is nice. So the first one that's come up is, can those using cloud and HPC for their research projects compare and contrast working with the two facilities and how they link together?
Shelley, maybe you could take that question for us.
Well, if you'd like, I can attempt to answer because we actually use both of the systems. Look, it's nice having the different variety of machines, because what you really want to do is to tune the work that you're doing for a particular architecture. So the scale that Gadi is able to run means that if you need to be able to run simulations of hundreds of thousands of atoms, it's a very highly efficient machine. And also for our own work, when we do stuff on the ATB, I'm actually Shelley's supervisor, if people didn't realize. This allows us to turn over molecules very quickly. Now, what we like about the cloud facilities, however, is that they're expandable. We can, when we have additional load, we are able to expand into the cloud, and so we don't generate delays and bottlenecks. And we can also tune the size of the job in order to be highly efficient on those size machines. So I really don't think you can consider one or the other. I think it's really a question of having a range of appropriate architectures. And so at the moment, we're actually pushing through more of our larger molecules on Pawsey because these actually take a wide variation in time to complete. Whereas on NCI, we have to make sure all of our jobs are all complete in a very similar window to use that machine efficiently.
Yeah. Certainly from my point of view, just looking now at what's available on Pawsey itself, I've got I'm on Nimbus, but we have the CPU and we have the GPU, and it really is very much horses for courses. Certain things just really do not work all that well on the CPU, but other things really do, and you wouldn't work at all well on the GPU. So it really is just a matter of having a selection of machines to do the available jobs that really best fit those jobs.
Cool. Okay, that's really good. I'm glad to see that the breadth of services and technologies that we can provide are all complementary and working together for the work that you're doing. The next question is how do you keep abreast of what's happening with the rapid changes and the large amount of research occurring?
It sounds like you've got an answer, Michael, to that.
I was going to be facetious and say you barely do. And that's the honest truth.
Yeah. Tom?
Yeah, same. There's a lot. Yeah. And then you've got the pre-print servers as well. So people are publishing on there, and half-baked jobs as well. So they can get them out really quickly, So yeah. As long as you've got an idea, you can put it on a print server these days. So, yeah.
I guess I have a corollary question then. It's, there's a lot of research happening. There's a lot of preprint publications that have extremely varied quality of work, right? If we're not waiting for peer review, we can't wait for peer review to get answers to certain questions. I mean, how easy then is it to gauge the quality of the work as stuff just popping up on the preprint archives?
I also do work in publication ethics. I'm also involved with the Committee on Publication Ethics, and let me just say that this is an issue that is very much challenging us in that organisation, because we have relied for all these years post Second World War on peer review, and not having it, whilst it's very imperfect, not having it at all it really is now back to buyer beware. And because we've seen the damage that can be done when a not particularly well done study is put on a preprint server, and is picked up by the press and totally misinterpreted in a tendentious way. So it is an issue, and I think the only thing you can be is a well-read scientist.
Mark, I guess we have tackled that locally within Australia, globally within Galaxy Project by reanalyzing every COVID-19 genome that is made published on the SRA or ENA as a nightly exercise, and Galaxy Europe's taking a large share of that work. But the publication I highlighted in my short presentation took some of the initial apparent SARS-CoV-2 containing whole genome sequencing samples and reanalyzed them, and were able to determine that a number of them had no viral material in them at all. So they had been tagged with metadata to suggest they had come from someone who was infected, but on analysis there was nothing in there. So yes, definitely buyer beware, because the metadata for those genome data sets suggested that they were useful. They were not.
Gareth, actually, that's a nice segue to a question here from Stacey. She's wondering how your work has impacted on your global partners and vice versa. So how has the genomics workflow impacted on other scientific workers for COVID and drug-related sequencing and targets?
Look, I think it's nice what you had in my talk and then in Shelley's talk, enabling talks. So we can't say we're doing that primary research, but we're very proud to enable that primary research and make the entry-level ability to trigger those workflows and those peer-reviewed workflows as easy as possible. So we track it right now by the number of uses, and we can say that that is showing a lot of reuse. What it has done as well, importantly, is because we have worked with our global partners in Europe and America, we know the workflows, the references, the tool versions underneath all that are identical globally. So when someone says they've done something on one of the Galaxy servers, then it is absolutely reproducible on the others, which should also help combat some of this sort of variety in analyses.
Yeah, thanks. I think I've got another question here from Lucy for Megan. She'd like to know, how does knowing about the movement of proteins influence the design of a drug to prevent viruses like COVID-19?
Okay, this one's going to be a visual one. So if you take a photo of me with my hand like that, you've got a picture of my hand, but it doesn't actually tell you how my hand moves. If we work out how my hand moves, then we can work out whether it can grip something, bind something, and from that we can use that information on the movement, how often my hand is open or closed, to work out whether we can put a drug somewhere on that protein. So that's, I think, knowing how things move, how they work, whether there's actually somewhere that you can put a drug, and how that drug has to be and how it has to interact with the thing it's binding to is really important in drug design.
Okay, I have another question, and it's from Lex to everyone. Do the presenters have any thoughts they would care to share about the COVID-19 research occurring on the World Community Grid platform? I think everyone's Googling that.
Yeah, pretty much.
That's a really hard one. I can't speak for the World Community Grid program, but one thing I can say is that I think that COVID has been an amazing opportunity for computational researchers to come together. We're some of the only people who are equipped with the skills to actually take their research home to their study, you know, to their kitchen table and continue on. And everything's been, well, we've been able to set everything up so that we've got remote access to supercomputers, and we're emailing, Skyping, Zooming with people all over the world to see what they're doing. So in terms of a global community, I think it's been an amazing opportunity for us. It's really brought us to the forefront.
Good answer.
I got another question for everybody. So we're in the position to do this work because of the time you spent the last 10 years working on molecular research. You can't just suddenly start work on a COVID virus, but you understand this work because of the last 10 or 20 years of research you've been doing. So to what extent or how did you end up doing this COVID work, you know, on the basis of your historical research? And I guess that's really open to everybody here.
So in our case, I can answer that. In our case, we've got a lot of history in lung pathology, so and many publications in that field. So we understand the pathology of viruses, especially the inflammation in the lung and the acute respiratory syndrome part. So that's how we come into it.
Can I answer that, Mark, in a really boring kind of way? So first of all, absolutely, Galaxy projects and some of its champion principal investigators, one of which unfortunately passed away earlier this year during COVID, were virologists and had a passion in this space. They also had the challenge in America of coupling, keeping Galaxy project alive with specific biology. So not a national infrastructure grant program, but a specific research topic, and virology was their topic of choice to pin Galaxy project to. Here nationally, we're extremely lucky to be funded by a variety of national institutes that don't tee us to one key research goal. So we're just providing the platform a bit more agnostically, and that means that I can't say I have come to this from COVID, but we can provide a platform that enables it. The short answer is yes, Galaxy project has a history in virology, and that's where we ended up in this space.
And as for my part, I've been working for quite a while now on Helicobacter pylori, which is bacterium. But many of the techniques that I've been using to analyze that, in particular the fact that it's very highly recombinant, far more than SARS. I've been able to transfer the techniques that I've devised over the last several years to run with this. So I was able to get up and running pretty smartly, and that's been a short project, rather long in the gestation.
I guess from my perspective, I've been working on membrane proteins for a very long time now. Funnily enough, the very last project that was part of my PhD was a collaboration I started with Stefan Brouwer, who's one of my collaborators on this project. And he wanted me to model the boat transporter. There was no sequence, there was a sequence, there was no structure, and we had to go at modeling it. And I've had a very loose collaboration with Stefan over the last 15 years now. And he has been working on boat because it's a good target for diabetes and other metabolic disorders. And we brought in, or Stefan brought in Mal McLeod, who's a medicinal chemist who's designed a whole lot of drugs. Mal said to me, can we test these out in the structure you made, a gazillion years ago? And lo and behold, in February this year, it turns out the boat is part of the COVID receptor complex. And so we actually had a wealth of information between the three of us to start this collaboration and really kickstart it with COVID. And then having fantastic postdocs like Katie and Steve also helped this a lot.
Yeah, so from my perspective, like I said, the focus of my PhD has always been on these fusion proteins. So being given the opportunity and the time from NCI and Pawsey meant that we could expand that out to something like COVID. So I already had the background in doing that. In respect to the ATB, the advantage of doing something like pushing all these phase 2 drugs through so that we can parameterize them is that it can be applicable to much beyond COVID. So now obviously our world focus is COVID-19, but there is also the potential to to test these drugs for things like influenza and other, important things that need treating. So I guess the focus there isn't specifically on COVID, but it definitely does have an impact in the pandemic.
Cool. Okay. I saw that Sean had raised his hand in the meeting. Sean, did you want to say anything or?
Yeah. Thanks, Aditi. I had to jump off for a bit, so my apologies if the question might have already been asked. Was for Megan. Megan, in your interesting summary of the work you're doing, I got the impression that there's a receptor, and you're thinking about a drug which could lock up that receptor so that it can't be accessed, if you will, by the virus. The question that occurred to me was, well, is that receptor there for some other natural purpose? And if your drug is locking it up, is that going to have some kind of knock-on effect on some other function?
Yeah, well, that's the interesting thing, because the receptor, you've got two parts of the receptor. You've got ACE2 and you've got BOAT1. So ACE2 is involved in blood pressure regulation, and a lot of people have been trying the drugs that are out there already, clinically approved drugs that target blood pressure regulation through ACE2. The other one, BOAT1, is, so the drugs we've been testing out are actually leads for anti-type 2 diabetes drugs. And so, so yes, there could be knock-on effects. I guess this is something that people have to work out. But everything does have a knock-on effect. I guess you've got to balance it out. I mean, as with the repurposing of many drugs, they're good at what they do, but you can have terrible side effects. So I guess, we're reducing the use of experimental techniques, trying to fast track the pipeline, but ultimately these things need to go to clinical trials as well.
Got it. Thank you, Megan.
Now I have more of a technological question from Ali, which is open to all the panelists. He asked that it would be nice if researchers could share the challenges they face in the computations, including data storage, code optimization, and visualization technologies and tools they use. So that's quite a wide-ranging question. It'll help us and NCI and Pawsey and others working in the technology space to set up our future workflows in ways that could overcome probable difficulties. Anyone want to have a go at that?
Is that not what Mark is doing with Galaxy?
Gareth, yep.
Gareth, sorry.
Yes, so we do face some of those challenges. Working with all you guys, it does help it. One of the mechanisms we face, and I know given some of the audience and questions here, is that we, at this point in time, structure our code to run on fairly generic deployed VMs. As you saw, just a standard ratio of CPU to RAM. And that is because the way Galaxy Project works at our very high level is to have those 7,000 odd tools very shareable across almost every deployed version of Galaxy globally. And that gives us that rapid deployment. However, what we would say is that as researchers are getting more ambitious, certainly the last 18 months with genomics research, we've identified the need that's already been spoken about highly here of having pulsars configured to be high CPU worker nodes or high GPU worker nodes. And that's what we're exploring, so that what we can do is offer tool sets and workflows that take advantage of that compute infrastructure.
Thanks, Gareth. Okay, I have another question, and this one's for Tom. Tom, leaning on what you've done previously, we'd like you to talk about your wet lab validations. How do you validate your findings from the computer in the wet lab with animal and viral models?
Yeah. So the steps. So for example, if we come up with a hit for the main protease, the easiest way to do it to start with is do an ELISA. So everyone's familiar with ELISAs, I think. And we can see if our hit protein is actually going to interact with the protein of interest. Right? So that's the first step. We'll do an ELISA. If that works, then we'll do an antiviral assay, an in vitro antiviral assay. And the cost increases exponentially as you keep going. So the ELISA would cost $3,000–4,000 for these new ones because they're very specific. And then for an antiviral assay these days, you're looking at about $20,000 to do a single compound. And if it works in an antiviral assay, then we'll look at we'll look at animal models. But it's even better if we can go straight to if we can repurpose a drug that's already used in humans, and then we can go straight to the human clinical trial.
Mm-hmm.
Yeah. So it's sequential, it's steps.
Yeah, right. Yeah. Thank you. I have another question here for Michael. Michael, so why does evolutionary history matter with these strains?
Couple of reasons. The first, perhaps more unfortunate reason is because of the is that science has come into politics. So politics has come into science. Sorry, and I wish it was the other way around. Politics has come into science, and we now have this debate about where the virus did come from, which is totally specious. But there it is, and that's quite clear from the evolutionary biology that this thing is its progenitors have been it's been evolving to be where it is, and it was just luck of the draw that. And misfortune that it happened where it did. I might also comment that although I've seen the SARS-like viruses in bats in China, and also the MERS-like viruses in bats in China, that's only because the Chinese bothered looking. So the issue is that there's not been enough publication of those sort of genomes around the rest of the world to really give us a full picture. And it's only by getting that information that we can actually go to the final idea, which is a one planet health. We have to start thinking in terms of those, this world is not just us and our own health. And again, the evolutionary biology gives you an access to that.
Yeah, good. Yeah, thanks very much, Alan. Tom, go. Tom. Oh, it's been a long day. Michael, right. Yep, got there in the end. One more question from Helen. Helen has a question for Tom. You mentioned 2 different docking programs, and I was wondering what the reproducibility is like between different softwares.
Yeah, correct. So one of them, the reason we try 2 is one's commercial, a very expensive program, and the other one is a free program. So we just wanted to make sure that both arranging it. And our R value was 0.7. So there's quite good correlation between the 2 programs. The one that's freely available, Autodoc Vino, that people will know about, and Schrodinger's other one. So yeah. The commercial one, the Schrodinger one, allows us to do batch. So it allows us to do quicker, more work. But in terms of validation, they're both pretty good, very similar. Yeah.
Cool. I think I've got one more question. Unless anyone else has some more, feel free to put them in the chat. And it's very generic and open to everybody. So how, maybe an easy one, how are computers relevant to COVID and medical research? So part of the context for that obviously is NCI has just gone through the deployment of Gadi, their new supercomputer. We're about to go through our transition as well, and we're really, I think, in the middle really of a real step change in the availability of high performance computing facilities for researchers in Australia. And we're hoping those will be well utilized. But yeah, I guess there's still work to do with, testing things on animals and what have you. So I guess how critical is the computer part of all this?
I'll give that one a...
Please, Megan.
Are you sure? Okay. I guess my view of it is experiments show you certain things. If you run a gel of a protein, then you'll see how big that protein is, but you won't be able to see what it looks like. You won't be able to see the interactions it makes, what amino acids match with the other parts. The same with the membrane protein. You can take a low resolution EM image of a protein in a membrane. It will tell you that the protein's in the membrane and how much sticks out. It won't tell you whether or not there's a phosphoinositol or a phosphocholine, or what type of lipid is sitting there, and whether a cholesterol is interacting, and how long this all happens from. You can't get this both atomic resolution and time-dependent interactions for my kind of work using experimental techniques. Now, the other thing is the time I've been given, or we've been given, for this NCI project, the COVID project. If I used my desktop computer at home, my son plays Fortnite. If he played Fortnite 24 hours a day, 7 days a week, it would take him 26.5 years to use the computing power we need for this COVID process. We're using all of that before Easter next year. So, that's an awful lot of computers lined up in a row to play Fortnite or whatever else. So I think they've got an incredibly valuable place.
The other thing I think is that the other part of the answer is to go the other way. And that is, there is nothing that I, or in fact any computational biologist will do, I believe, that ultimately all we do is provide hypotheses, models, things that could work, and then we have to narrow it down. So it's this, we have to work hand in glove with the experimentalists to test out the specifics. But experimental biology is getting ever more expensive. Computational biology is becoming ever more cheap. It's clear which, how the way forward suggests is to do the computational biology, which then suggests experiments for the biologist to try and turn into drugs. And particularly when you start moving into clinical trials, the cost benefit is just way, way, way obvious.
I agree with Michael as well. And just to give you an example, with our library that I showed you, we've got 800 compounds. In order to make those 800 compounds, to synthesize it, we need about $5 million. So it's impossible to even contemplate doing that in the lab. We have to screen using computer-based stuff. Yeah.
Can I make a comment? I've got to make 2 comments. And first of all, I've got to say, Megan, I suspect there's a number of people up for that amount of fortnight challenge. But that aside, I was actually going to say the opposite to what Michael and was just commented on as well, which is that a lot of the data generation we're seeing has gone incredibly cheap, and researchers are getting larger and larger ambitious projects, particularly in genomics and proteomics and metabolomics. And I think we almost have a role to support that by enabling them to not bottleneck at a large amount of data files, but turn that into real results. And that's what Pawsey, NCI, Biocommons and Galaxy, all these services do.
Great, thank you all very much. I do have one last question from the floor, and it's from Aditi. Aditi has a question from Michael. The painting behind you, is it some sort of sequence or just a coincidentally relevant to the conversation painting?
No, it's actually an Aboriginal artist from the northwest of Western Australia called Clifton Mack. It's meant to be, it's representing the water on different rocks after rain.
Wow, it's beautiful.
So,
Aditi, I think, so thanks very much to all the panelists. I think that was a really, really interesting set of questions and a very lively conversation. I think now Aditi is going to take over and take us through our networking session.
Yes, so again, like Mark said, thank you all for coming together today and doing your presentations as well as this Q&A. It was really, really, really interesting stuff, and I feel like I know a little bit more about the COVID-19 research that's going on, especially in Australia and around the world as well. So thank you all. So that kind of ends the formal proceedings for today's event. You're all welcome to stay in this meeting room and talk to each other now. It'll be a little bit more of an informal setting where you can all just have a chat and log off whenever you need to. So yeah, thank you all. Just to wrap up, I'd just like to let you know that like this event, there are a few others that are on the Pawsey's website at the moment. Our next event, our next Pawsey Friday in August focuses on quantum computing for industry. So please feel free to go to our website and register for that event if it's relevant to you and your work. You can also use our website to sign up as a Pawsey friend, where you'd get notified of our events, our training initiatives, and some of the cool work that we're doing at the Pawsey Centre. And if you've missed any of our webinars, our training sessions, or these sorts of events that we've had in the past or can't make it for our future ones, they're all uploaded to our Pawsey YouTube channel as well, which is youtube.com/Pawsey Supercomputing Centre. So thank you all, and yep, I encourage you to stay on and have a chat if you need to. Otherwise, hopefully I'll see you at the next event.
Yep.